So indeed, I think it might be the first neuro today. And we're going to talk about a property of stem cells that is shared across systems and that we've heard about today, which is quiescence, we've heard about, but we're going to delve a bit deeper. And just to put it a little bit in context, in a nutshell about my lab, we're interested in size control, which boils down to often to stem cell properties. And particularly in the CNS, this is intimately related to cell type specification, proportions of cell types, so their identity. And we're interested in this. We have so far focused mostly on development and cancer, but in its wider context. And in cancer, particularly drivers there. So this is the issue of quiescence. It's a binary decision whether the stem cell commits to the cell cycle or not, particularly commits to replicating its DNA. However, it's a threshold property and becomes binary from what is in fact a graded series of states. And so we refer to quiescence depth according to if the cell takes longer to get back into cycle or shallow if it takes a shorter time. And what is quiescence? It's defined as reversible cell cycle arrest accompanied by diminished biosynthetic activity, particularly lower levels of protein translation. And it seems to have evolved because it's a protective state. So because there's no DNA replication, a certain type of mutations are prevented. It also is a state where protein is often levels downregulated, so cells are oblivious to a lot of stimuli and insults. Now, this is advantageous to keep the cells pristine for these useful functions here, and particularly in the CNS, there's a lot of interest because it's not just this homeostasis and kind of matter of cells to make up a certain mass of tissue, but there's also higher level cognitive and mood properties that seem to be regulated by it. And in cancer, it's a problem because its protection means that the insults we normally use to attack these cells, be it radio, chemo, or even immune therapies, are often, the cells are oblivious to them and then can reconstitute the tumor. So, starting from this very humble model system that many of you will be familiar with, neural stem cells here too have a phase of quiescence between pre- and post-embryonic stages. And we use mainly two assays in the lab to look at this process. One is precocious or delayed activation around this normal physiological process, but another is anachronic re-entry into quiescence at late level stages. And today, actually, a lot of the results we'll see are from this assay here. Now, interestingly, the cells change morphology when in quiescence or active states in neural stem cells. This is in the fruit fly. They have a fiber, which may remind you of the radial glia fiber that you're used to seeing in mammalian cells, which they don't in active cells. In this talk, we're not going to address the function of the fiber that we're very interested in, and there's a lot of ideas around, and it may have been multiple, but we're using it as a marker. And in mouse stem cells too, at least in culture, they also have a more complex morphology when quiescent than when active. We found many years ago a mutant that at late larval stages, when these neural stem cells are always like blobs and they're active, had also this fiber very reminiscent of when they're quiescent, and they were always invariably pH 3 negative. So we wondered whether this was quiescence. And long story, we mapped it, we identified the lesion responsible, and we were able to phenocopy it by RNAs for that target gene, where you can see here this ectopic fiber at late stages. And I'm going to introduce you briefly to how we quantify this phenotype. Bear with me for a second, so that it will then be very easy for you to take in the results of some graphs I'll show. So in the fry CNS, each brain lobe has around 100 neural stem cells, which is very convenient, so we can count them and immediately get percentages. And they are here labeled with a deadpan, which is a HES5 family transcription factor. There's roughly 100, and 20% odd are PHC positive at any one time. During quiescence, which is the early larval phase, they are all still there. We know that from lineage tracing, but half of them are invisible by deadpan positive staining because they downregulated so much, but we know they're there, okay? And half of them are visible by deadpan, but even few are visible with this other marker, which is an actin binding protein called Miranda. And only when you have this cortical marker do you actually see the shape and the fibers. So very few we can see the fibers. But again, as I say, for GFP and nitrogen, you can see they're there. And only a very few number that never enter quiescence, which are about 4 per grain load, are PHC positive. Okay, so this is the general picture of quiescent active stem cells in the normal wild-type organism. Now, when we had this mutant with the ectopic fiber at late stages, you can see that there's some similarity to what would be a normal quiescent, but bear in mind this is late stage. The homozygous and hemizygous ovarodeficiency have a similar phenotype, suggesting this is a strong or null, a strong hypomorph or lung, or null, a strong loss of function. Because Miranda was expressed late, it stays on in the cell, so it paints more of the fibers. I hope this is making sense. And then the RNAi is somewhat of an intermediate phenotype. So the question became, is it really quiescent? Are these invisible cells really there like a normal quiescence, or have we induced differentiation or even cell death? Okay? And before I address those very important questions, I'll just introduce you to what this gene is, because at the time it was simply a gene number. And it's a family of conserved, but in a very broad stroke, because they're natively destructured and so quite at primary structure level, quite a divergent set of proteins that are FG nucleoporins. And these are components of the nucleopore complex, as you know, is the gateway between the nucleus and the cytoplasm. And again, very briefly, just to put you in context, there are many nucleoporins that constitute the pore. There are about 30 types of polypeptides, and they're in an eightfold rotational symmetry in two kind of donuts on the inner and outer membranes of the nuclear envelope, and then some that are exclusively on one side or the other. So we have different kinds of stoichiometries per pore. And this novel nucleoporin that looks like a nucleoporin is most closely related to one called NUP98, which is actually found on both sides of the pore, so potentially a NUP98 too, let's say. So back to the question of, is it really quiescence? Did we induce those cells to become something else, or are they really still there? So to do this, we induced RNAi that I have told you phenocopied that mutant phenotype, albeit with a weaker expressivity. We induced the RNAi using the alpha-UAS system that some of you may know is temperature dependent. So if we up the temperature, we're inducing RNAi. If we lower the temperature, we stop the induction and let the cells recover, which is what R here means. So we induce, recover, and analyze the phenotype just after induction at different times, and after we let or don't let recovery, okay? So these are controls for a cherry RNAi that has no phenotype similar to the wild type, and this is the RNAi at 31 degrees, so when we really push it, and here's when we induce and recover, or induce for longer and recover. And so you can see that you start seeing the RNAi phenotype a bit stronger with this longer time duration of induction, and that there is a significant recovery. Of course, we don't have a lot of time for the recovery, because meanwhile the animals pupariate and actually neural stem cells disappear because they actually do differentiate. So the phenotypes are milder, but they're reversible. That's very important as a definition of quiescence. We saw a similar result when we induced RNAi of the related nucleoporin 98, actually even stronger. So it does seem by all possible functional assays that it's quiescence, and there are no quiescent markers for any cell type on any tissue in any organism. So we have to go functional really to define that it is quiescence. And we asked whether this quiescent-like phenotype was specific to these nucleopore 98-like molecules, or was it more widespread, seen if we downregulated components of the pore? And so here they are in these background color-coded as the subclasses of pore constituents, as this cartoon. And what you can see now, you know how to look at these graphs really quickly, is that with many RNAis for some nucleoporins, we got phenotypes, with others we didn't. But across classes, there were some nucleoporins that had a phenotype. So it's not specific to a subclass or an anatomical subclass, if you like, within the pore. It is pervasive throughout the pore. So this suggested to us that it might be a nucleocytoplasmic transport function, as may not be too surprising to you, but in fact, many nucleoporins moonlight as other functions in the cell. Some act as transcription factors or chromatin remodelers. Others, in fact, even regulate microtubules, and I told you that there's a morphological change in these states. So we were interested in really nailing it, whether there was altered nucleocytoplasmic transport between these states. And another way to go about it was to look at, other than nucleoporins, other components of nucleocytoplasmic transport, particularly of active transport, which you may know is fueled not by ATP but by GTP, and there's this cycle of GTP, run gaps and run gifts that fuel it, and then there's importins and exportins that are adapters that bind to the cargo proteins to go in and out. So we performed RNAi on those. And again, now you know how to look at these graphs. In fact, here the result was much more specific and led us to very interesting places. So many of the importins or exportins had no phenotype across multiple RNAis, including ones that we know from other contexts actually work to deplete the target protein. But we have some very strong phenotypes with RAN and its RAN gap and RAN depth, but also with specific types of transportins, which is the common name for these. And what is now emerging is that these adapter proteins transport functionally related cargo. And looking at pathways that they seem to transport in and out of the nucleus led us to become very interested in RNA metabolism and RNA localization. And we started by looking at polyaRNA with a simple oligo-DP probe. So let me take you through what this is. This MB is a mushroom body neuroblast, which is one of the few I told you, neural stem cell that exceptionally never undergoes quiescence in the fly. So they're always active, okay? And in the ventral nerve cord, at early time points after larval hatching, they're deeply quiescent, and then they start to emerge from quiescent. They still are halfway there, they still have a fiber, until eventually they grow out of this state, become active, and lose their fiber. And what you can see here is a diaphragm that labels the nucleus, and these are a few optical sections, so it's not always terribly clear, but we performed this on a few numbers and are quite confident about these results. You see the cytoplasm has a lot of mRNA, and there's even quite a bit in the nucleus towards the nuclear periphery in this active cell. And here in this quiescent cell, there's less RNA. That is not surprising. We know that quiescent cells transcribe less in addition to translating less. But what is interesting is that there is some strong puncture within the nucleus. I don't know if you can see. And as the cell exits the cell cycle, now you become the cell expresses more RNA, lots in the nucleus, more out in the cytoplasm, and eventually the nucleus clears up again. And so when we quantified the nuclear to cytoplasmic ratio, what we saw was that the active, the permanently active neural stem cell has a very low nuclear cytoplasmic ratio because polya is mostly cytoplasmic. The deeply quiescent cell has a very high nuclear cytoplasmic ratio, so less RNA or less polyaRNA, but most of it in the nucleus. And as the same cell that we now follow over time exits this quiescent state, the nuclear cytoplasmic ratio decreases again. When we artificially induce this quiescent-like state by RNAing a few nucleoporins, our novel one, or the NUC98, we also see some instances, in fact, in which the nucleus is in fact screening with oligo-DP. And this protein RANDA that I told you was cortical and actin-bound is in fact in some instances found in the nucleus, which actually we know is the case in the normal deeply quiescent cells. So is this a quirk of the fly and of neural stem cells, or is it evolutionarily conserved? So in our lab, we also make use of a mouse model that we obtained, in fact, from François Rimon's lab, that many of you will know here, and which are adult hippocampal neural stem cells, primary cultures that are shifted into quiescence by BMP4. And so we looked at these cells very extensively in our lab and François' lab in vivo in the mouse, and we saw the same thing, that there is, now we have lots of cell numbers, the cultures are monoculture and they're synchronized, so we can look at many cells at once. There's less nuclear polyaRNA, there's less cytoplasmic polya in quiescence, but there's much less in the cytoplasm, so that the nuclear cytoplasmic ratio again is higher in quiescence. And interestingly, we saw actually very similar ratio in vivo. Of course, the spread is less here. Here we have no idea how long the cells have been in quiescence, whereas here we can actually regulate the depth of quiescence by the length of exposure to BMP4 we showed. So it pertains to neural stem cells in vivo and in vitro, in fly and in mouse. I don't have time to show you, but we saw this is the case also. William provided Alice Rossi, in ours and François's lab to look at hematopoietic stem and progenitor cells. It holds there too. We started to look still preliminary in muscle satellite cells. It holds there too. And Alice also looked very extensively at glioblastoma patient cells. We now have looked at 3 different patients. So quiescent cells seem to, maybe universally, but quite prevalently, retain some poly-RNA in their nucleus. And most poly-RNA, as you may know, is messenger RNA. Alice also showed that downregulation of NUC98 in the mouse cells, I'm not showing the reversibility here, but also induces quiescence, and that this is reversible. And we were now interested in a few questions that stem from here. What are these mRNAs? Is this a physiological mode of regulation, or is it a quirk that we found in this screen? Although having noticed altered nucleocytoplasmic partitioning in a variety of cell types in quiescence, it seemed likely that altering the nucleocytoplasmic regulators would indeed be a physiological mode of regulation. So Alice performed proteomics now on the mouse cells, because that's another beauty of having this monoculture, is now we can do omics very simply. And we see proteins that are upregulated and proteins that are downregulated in these cells, some expected known proteins of the 2 categories we saw. There are more proteins we know that are downregulated than not, which is one of the difficulties of having no markers of quiescence. We found those categories associated with each of these that we can discuss later. But to the point of what we were asking here, most nucleoporins, most active transport components, and mRNA export components are downregulated in quiescence. And we also asked about splice factors, because we had at the back of our mind, why is mRNA being retained? Because mRNA is not exported by the bulk export pathways, it's not exported by exporting. In fact, it has a dedicated pathway, a TREX complex mediated export. However, if it's not completely processed, it is retained, and one of the key processing steps is splicing. So we wondered whether the RNA would be less spliced, and I'll come back to that. Intriguingly, we found that actually some splicing factors are upregulated, although the majority are down. But this might be perhaps commensurate with downregulation of transcription. And the next key question, of course, is what are these transcripts that are retained in the nucleus? So we fractionated these cells into nuclei and cytoplasm and performed RNA-seq. However, RNA-seq on these fractions. And what we're plotting here is not necessarily the exact transcript abundances, but what we're calling a Z-score, which is the fraction of transcript that's in one compartment versus the other. So a positive Z-score indicates nuclear accumulation, and the negative Z-score cytoplasmic accumulation. And this is a logarithmic scale, and so the numbers are on the low scale, but you can very meaningful. And when you look at the median here, you can see that upon 3 days of the MP4 exposure, so shallow quiescence, and then more so upon 10 days of the MP exposure, which is deeper quiescence, the median goes up. And it goes up significantly. And what we found, crunching all these numbers, is that out of maybe, let's say, 30,000 genes that we find consistently expressed in these cells, at least 2,000 are nuclear retained to a great level, and when I say great, means a 4-fold increase in the proportion in the nuclear structure to the cytoplasm, which is very significant. Also importantly, this has an impact on protein expression. So with about 80% or more prediction ability, Z-scores that are positive, so transcripts that are held in the nucleus, are downregulated as protein, and conversely, transcripts that increase their cytoplasmic expression relative to nuclear in quiescence are upregulated at the protein level. And this explains why, as many of us, we use RNA-seq as a proxy for what proteins the cells really have, and overall at the population level, it's a good proxy, not at each individual, because of course post-transcriptional regulation is important, but even that population level proxy is completely abolished in quiescence, because we have a lot of transcripts that are expressed that are not converted into proteins. So this correlation is completely lost, and the two things are uncoupled. So we found also interesting categories of what's held in the nucleus, and we think there's a hierarchical deployment of these factors according to the depth of quiescence. And we also looked at how processed they were in terms of splicing. So we sequenced with enough depth to look at intron retention. And to our surprise, we really had to confirm this. Most proteins that are held in, and most transcripts that are held in the nucleus are actually more completely spliced than not, than in natural conditions. And we retrospectively make sense of this by thinking that because they're steady state means that they're longer in the nucleus, they have more time to be accessed by processing factors. And so we have a large category of factors that have lower intron retention, most of the ones that are held in the nucleus, but a few that have actually higher intron retention. And these are ones that are largely involved in outside regulation and regulatory and so we think that there's a hierarchy of deployment of factors according to whether they're not held in the nucleus, or held in the nucleus, very processed, ready to go, or held in the nucleus, unprocessed, still need to be spliced, or the ones that you still need to go all the way back to transcription, so that you have a stepwise then reactivation, or conversely, a stepwise entry into quiescence, which explains or describes a bit this graded series of states, that is quiescence. So in summary, we'd like to lay down on the table this important post-transcriptional mechanism of regulated quiescence. We don't know yet why they are retained, and the fact is that we don't think it's the bulk mRNA export, because we don't see all transcripts held in this way, and there's very little known about selective mRNA export pathways, so that's one of the things we want to go into next. Alice is also looking at other nucleoforming functions, mainly in regulating gene expression. We have evidence for different structural organization of the pore in quiescent and active cells. So lots of very interesting cell biology that we want to go to next. That in the interest of time I won't go into. What I've told you about today is on bioRxiv. Some of the data isn't, for example, glioblastoma data isn't there. And I'll be very happy to talk to you if you want about this or where we're going next. I'll summarize, hoping that I'm still on time, that quiescence alters the concentration of nucleocytoplasmic transport factors. We don't know yet exactly what this means for function, but we know that the degradation of these programs can induce quiescence in both biomass and cells. We know that there's strong nuclear bias of a subset of transcripts, very strong nuclear bias of a subset of polyadenylatedtranscripts RNAs in a variety of quiescent cycles. That this increases the quiescent depth, and that this has a functional impact on protein expression with high concentration of stability, and that the processing of these transcripts are retained is also different. Most of it is ready to go in terms of splicing. Others have that with higher intron retention. And indeed, we've seen this in multiple types. So we think this may be a universal property of quiescence. So I'd like to thank lots of people involved in the work, both at King's and in Pettit, and particularly Alice, that has been very heavily involved halfway through the project, and she's represented in both. Previous lab members and also collaborators with King and here, you know, several people, are funding for this project.